See, through the whole week, we have touched upon a lot of chapters, lot of concepts, everything has been touched upon. I feel better to stop it at that till what we have done till yesterday. Not add on more concepts in terms of elaborate discussion. What I want you to see or what I want to show you today is how I make the syllabus map. Every level of notes making that I have told you, I will show you how I make it after you have attempted it and struggled with it and made it by yourselves. Alright? And with a little bit of gap, you are supposed to complete syllabus map by 31st of July 2024. Correct? No, 31st of July was the time by which you were supposed to complete. Most of you have completed by now at least. Anybody who is yet to complete syllabus map, raise your hands. Anybody who is yet to complete syllabus map, all of you have completed syllabus map. Good. A good syllabus map is quick revision tool. How should it look? I will show you. How a good syllabus map should look, I will show you. So that will give you a reference of how to look at it. Why would I not give this to you right away in print? You will be able to make sense of it when you have done it. If you have not done it, you will not be able to understand the nuances of it. Alright? So I will be giving you the syllabus map for both 11th and 12th, all the chapters in a proper file format. I will share it by the time I come in the next visit. Next visit I will be coming on September 7th only. So in a very short period I will be coming. By then you should be completing your level 1 notes also. After you complete your level 1 notes, I will give you my level 1 notes. Soft copies. Your notes, your revision notes should be hard copy. Handwritten. What you have written. Don't copy exactly what I give you, but use what I give you as a study tool and try to build your own personal notes, handwritten notes. That will be your revision tool. Alright? Now as I take you through the topics and subtopics in the syllabus map, I want you to recall. Let's see how much of recall can happen. See, this is the list of chapters. We should know it. The small things are extremely important. You should be able to tell the list of chapters in a sequence. How many chapters are there in class 11 revised syllabus? 19. Class 12? 13. So we have all the chapters in a sequence. 19 chapters in class 11 and 13 chapters in class 12. Some of the chapters which were earlier it was 22 and 18, 22 and 16. 22 and 16, totally 38 chapters were there. Now we have 19 plus 13, 32 chapters. 6 chapters has been reduced. Have the concepts gone away? Have the concepts gone away equivalent to 6 chapters? The concepts equivalent to 6 chapters have not gone away. They were all present in these chapters in different forms. So the repetitiveness has been eliminated by removing certain chapters and pushing the concepts into these chapters. Alright? This is the mapping that I do. Serial number is just to indicate the chapter number, topic, subtopic, concepts or subconcepts, some details if at all you want to elaborate a little bit more detail into that, add something else, or if you want to put down some 1 or 2 examples there, you can use it appropriately. And reference, which chapter, which page number, reference from your NCERT book. So when you are going through the syllabus map is your study material, is your revision material. You are looking at something, a particular concept, you are not able to recall. All of this, when you look at it, it should have a recall value. What is living? You should be able to tell something in your mind. Defining characteristics of life. What are the defining characters? The point should come in your mind. Alright, here it's given already. Growth, reproduction, metabolism, consciousness. Levels of biological organization. Cellular, tissue, organ system, organismal. What does each mean should be a recall for you. If you are not able to recall something, You should not be wasting time going and searching for where it is in your book. A small reference here telling you which page number it is present. Which page number it's present in the NCERT book. So the reference is to NCERT textbook basically. Syllabus map is referring to NCERT textbook. All the linear notes you make should be with the reference to NCERT book. Linear notes should not be going to various sources. All the books in your library, all those big books that you are carrying, those should not be the reference for your linear notes. From that, you should be picking up relevant things based on what questions you attempt. So additional information should all be a part of your post-test analysis notes and not your linear notes. The entire reference for linear notes should be NCERT. Alright? Keeping it simple is very, very important. So let us see. What is living? Quickly, you should just go on telling. What is living? What is living? Tell it in your own ways. Which has life. Then the question will become, what is life? That which is present in the living. Where is your house? Next to my neighbor's house. Where is your neighbor's house? Next to my house. Alright. Where are both of your houses? Next to each other's. So, what is living? Can I say anything that is living is that which responds dynamically to the surrounding changes? Can we say that? Anything which responds dynamically to the changes happening in the surroundings is living. That definition you will not get it in the books. But will it not allow us to understand? This will also respond. If there is heat around, this will also get heated. It is also responding to the surroundings. But is it reflecting the surrounding changes or is it countering the surrounding changes? It is reflecting the surrounding changes. When there is heat, some metal will get heated up. So the heat in the metal is a reflection of the heat in the surroundings. But if the temperature around is very hot, your body also will get heated up. But the heat in your body is not just a reflection of the heat outside, it is a counter to the heat outside. It is countering the heat outside and trying to adapt itself to, keep the heat in a level of manageability. So life or living organisms, anything that is living will respond to the changes that happens in the surroundings dynamically. It will not just reflect the change, it can counter the change also. Right? Characteristics of living: growth, reproduction, metabolism, consciousness. What is consciousness? Do all organisms have consciousness? Do all living organisms have consciousness? For that you need to know what is consciousness. Consciousness is the awareness of the changes, awareness of the changes happening in the surroundings and active response to that. Ability to respond actively to that is consciousness. So does the bacteria respond to the changes in the surroundings? Can a bacteria respond to changes in the surroundings? It does. But is the bacteria responding based on its genetic predisposition, or is it responding by thought? Is there thought in the bacteria? There is no thought in the bacteria. Is the bacteria actively responding to it, or is it biologically responding to it? It is biologically responding to it based on the chemical awareness that it has. There is no consciousness in bacteria, or consciousness is very limited. We can say very limited consciousness. It has awareness. When there is a metabolic response,
that also can be considered as consciousness. In Sanskrit we say Jada and Chetana. Jada means Chetana and Dharona, no? Sisters, twins. Okay. Chetana means conscious. Chetana is consciousness. Jada means unconscious or with no consciousness. This used to be a Chetana Vastu, Chaitanya Vastu. It had consciousness when it was living. Now it is unconscious, non-living, Jada. Alright, those are the characteristics of living. So next what we are learning here, levels of biological organization. How do organisms exist in what levels? Cellular level, tissue level, organ system level and organismic level. Okay. Then biodiversity, definition and importance. What is biodiversity? What is the importance of biodiversity? Variety of species, genetic diversity, ecosystem diversity. Alright. Each one should ring a bell in your head. When you look at this, you should be able to tell what it is. So the syllabus map can be your pastime. You can use that and ask your friend, what is this, what is this, what is this, what is this, tell me what this is. That can be a nice way of asking each other what you are supposed to know. Alright. Need for classification, evolutionary relationships between organisms, phylogenetics, cladistics, species, basic unit of classification, genus, group of related species, family, group of related genera. Some examples given in NCERT only. So there is nothing further to elaborate here. So add some examples there into that column. Order, groups of related families, class, groups of related orders, groups of related classes is phylum, highest taxonomic rank is kingdom. So now this is the summary of the taxonomic categories that is given in the first chapter. Many times questions have come to asking you to arrange the taxonomical order. Taxonomical order arrangement itself is a question. 4 marks for this. Can you, I'll just take it away. Anybody tell me the taxonomical hierarchy in an order? One of you will stand up and tell. Those who feel confident of the taxonomical hierarchy in an order, starting from species, stand up and tell. See? Yes, can you? In an order. Right. Species, genus, family, order, class, phylum, kingdom. Correct? That's all, simple. See, what you have to know in a sequence is what the syllabus map will help you. Even if you get 4 or 5 questions in this order related things and simple recognition things. I told you, your entrance exam is a recognition test. It is a recognition test. Learn to recognize. For recognition, you need to have familiarity. Syllabus map will tell you what are all the things that you should be absolutely familiar with. Alright. Knowing what is the bare minimum is the key to success. What all can you learn? There is no limit to it. There is no limit to what all you can learn and accumulate, but there should be a limit to what is the basis, what is the basics, what is the fundamental. So, this will help us define the fundamentals. Then taxonomical aids. Again, questions come on the basis of the difference between these. We had one question on this in the first paper only. Correct? 2013 NEET paper, we had a question on this. Herbarium, botanical gardens, museum, zoological parks case. Because there are no subtopics in the first chapter, it is just recognition, what it is. I have just given you what it means. That's all. Botanical gardens, live plant collections. Museum, collection of preserved animal specimens. So do we have plant specimens in museum? No. Zoological parks, protected areas for living animals. Keys, identification of organisms. Dichotomous key, artificial key. There are two types there. And in museum, natural history, natural museum, history museum, science museum, some examples are given as per NCERT. Now what is equivalent to a zoo for plants? Botanical gardens. Botanical gardens are for plants what zoo is for animals. Correct? So those kind of questions can come. If you have to read this chapter from NCERT book, it will take you some 10 minutes. When you have transformed the book into a syllabus map, you are identifying all of that. When you look at something, you should know what it is. If you don't know what it is, you have the reference of where it is. Go back and just brush it up and come back. Every time you don't have to go back to your textbook. You can do all of this. You have already done this. Now you are making it in the form of syllabus map, but otherwise also during regular studies, you will mark it in your textbook. When you mark it in your textbook, you will not be able to establish connections between topics and subtopics. You are just marking, that will be random. And every time you have to revise it, you will have to take the entire book. But syllabus map, it will be about, if you consider the entire 11th and 12th standard syllabus map, properly edited and all if we do, it will not be more than some 80 to 100 pages. Okay. Not more than 80 to 100 pages of syllabus map. That 80 to 100 pages, see, if you look at the entire material that you have to study, NCERT textbook is about 11th and 12th put together 700 pages. 600 to 700 pages of properly drafted content. You have read it, and if you can bring it to less than 100 pages of outline, blueprint, it is good, no? And quickly you can revise. That 100 pages you can revise in 2 to 3 hours. Once you are familiar with it, as you keep doing it again and again repeatedly, as you keep going through that repeatedly, the same format keeps getting revised repeatedly. You should not be taking more than 2 to 3 hours or maximum 4 hours to revise the entire syllabus map of 11th and 12th biology, one chapter. If you sit for a day, physics, chemistry, biology, all the 3 subjects, you should be able to revise the fundamentals of all the 3 subjects in one day. So this is a very important revision tool. Alright. So don't ignore the linear notes that I have told you, keep making it. So that completes the first chapter. Chapter 2, biological classification. What do we study? In biological classification, the 5 kingdom classification. Subtopic, Monera. In Monera, okay, we'll just look at the subtopics. What are all the subtopics we will study? Monera, Protista, fungi, plantae, animalia, and viruses, viroids, lichens, all of that we will study. So those are things which do not fit into any of the 5 kingdoms. Viruses, viroids and lichens, they are all complex, and viruses are partially living. So now you try to recall, if you can recall this. Monera, bacteria, types. You can further divide, you can further make each one of these into different cells, so that one is in every cell. I just put it off in one cell. Bacteria types, Archaebacteria. Archaebacteria are methanogens, halophils, thermoacidophiles. This is a question, this has come multiple times. Which of the following are not Archaebacteria? All these 3 will be there, and one other thing will be there, like methanogens, halophils, thermoacidophiles, mycoplasma. These four are given. Which of the following is not an Archaebacteria? It should be mycoplasma. Alright. So eubacteria, Archaebacteria and eubacteria types, Archaebacteria and eubacteria. Eubacteria, cyanobacteria and mycoplasma. Blue-green algae. Cyanobacteria is also referred to as blue-green algae. Over bacterial structure, what all should we study in the structure of bacteria? Cell wall, plasma membrane, flagella, pili, ribosomes. Alright. There are some specialized organelles also for bacteria. What are those? Mesosomes. Okay. Nutrition in bacteria, autotrophic nutrition, heterotrophic nutrition, both are present. Photosynthetic autotroph, chemosynthetic autotroph, saprophytes and parasites. So here when you look at it, just looking at it, it should strike your mind. Are flagella and pili, are they coming from the cell wall? Are they coming from inside? What is it? Are they just attachments or are they extensions of the bacteria? You should be able to get a picture of how a flagella looks, how a pilus looks. You should be able to correlate with the diagram of that in your textbook. If you are not able to, go to that page. You will find it. You understand? So wherever you are not able to correlate, you should be able to cross-reference and quickly learn it. So okay, nutrition, photosynthetic autotrophs, chemosynthetic autotrophs. What is the difference? Autotrophs are those which produce their own food. What is the difference between photosynthetic autotrophs and chemosynthetic autotrophs? What?
Photosynthetic autotrophs use light, whereas chemosynthetic autotrophs use chemicals instead of light.
For what? For food production. Huh? Now with autotropism, can you re-articulate what an autotroph does? Do we say photosynthetic autotrophs use sunlight for making their food? Can you get more specific and give me a more correct response to that? What is photosynthetic autotrophs? Are autotrophs manufacturing food using the external energy source? What are the autotrophs doing? Photosynthesis is correct. Photosynthesis is a long process. Are they manufacturing food using sunlight? If we consider photosynthetic autotrophs or photoautotrophs, are they manufacturing food using sunlight? They make high energy compounds. So the external source or the inorganic sources are used for making organic sources of energy. The inorganic sources of energy are used for making organic sources of energy. And that organic source of energy is used for producing organic compounds within the living cells. We also use external chemical source of energy. But what we use to make our high energy compounds are not coming from the inorganic world. They are coming from some other living organism only. That is why we are not autotrophs, we are heterotrophs. So photosynthetic autotrophs and chemosynthetic autotrophs, the difference is photosynthetic autotrophs use the sunlight as the source of energy for making ATP. Let's say we will say ATP, high energy compounds, ATP, NADPH, all of that is made. Chemosynthetic autotrophs will break down some inorganic chemicals and use the energy released by the breakdown of inorganic chemicals to make ATP within their body. Those are chemosynthetic autotrophs. So photosynthesis and chemosynthesis, both are autotrophs, autotrophic mechanisms. What is the difference? You should be able to know. What are saprophytes and parasites? Difference between saprophytes and parasites? Okay. Parasites lives on living organism. Again, you have to re-articulate it. Saprophytes derive their nourishment by breaking down dead and decaying organisms. Draw their nourishment from dead and decaying organisms. Parasites draw their nourishment from living organisms. Alright. So we also use dead plant parts. Are we saprophytes? Tiger kills a deer and eats it. So tiger is eating dead deer only. Is tiger saprophyte? Then by the definition that we have given now, tiger, human beings, all of us can be considered saprophytes. Why are we not saprophytes? How do you differentiate that? If eating dead organism is saprotropism, before you eat anything, it is dead only, no? I was not well for the entire week. I am not well now also. I successfully survived the whole week on medication. Okay. How are we? Why are we not saprotrophs? See? You just need the syllabus map. Syllabus map and the right questions. Why are we not saprotrophs? Come on, this is fundamental, basics of basic. No confusion with parasites. What parasites eat from is not killed. Mosquito is better, it will just suck a drop of blood and go. We have to kill and eat. Mosquito can be, in the mosquito educational institutions, it might be going on like that. We are very kind people. We only take what we want, unlike human beings who kill everything before they take. And all the mosquito students will be like, oh, you should be careful from humans. We should innovate, we should do some innovation to ensure that our people are not killed by humans. Who knows what's going on? All right. Saprotrophs do not take food. You are a cricket player and you are a bowler. You are a bowler. You are playing against a you are bowling against a batsman who is extremely good with short balls. You give him a bouncer, he will hit it for 6. Short ball, striking short balls is the strength of the batsman. As a bowler, do you decide to get him out by bowling short balls? What do you want to do? That is what you don't want to do. You don't want to make the batsman play short balls. You don't want to play to the strength of your enemy. Correct? Playing to the strength of the opponent is making your fight very difficult. If you play to the strength of your opponent, then your chances of winning becomes lesser. Alright? Now I tell you why this antibiotic resistance has become such a big menace. Antibiotics are produced by bacteria to kill other bacteria. Antibiotics are produced by bacteria to kill other bacteria. It is a chemical weapon bacteria have mastered to ensure their survival in their surroundings. If I am a bacteria in some water body, I have my own colony of my own people. Resources are limited. If there is another colony of bacteria which wants to use the same resources, what will happen? There will be competition. And how do I win over the competition? With competition will bring in struggle for survival. And in the struggle for survival, there will be an attempt at eliminating each other. I will try to do something which will eliminate them. I want to kill them. I want to make them run away from this place. Or I want to kill them. They have to either leave the place or I will kill them. That is brute nature. That is brutal natural forces in play, struggle for survival. So in this process, bacteria have developed the ability to produce antibiotics. Antibiotics are the weapons produced by bacteria to kill other bacteria, so that they win the struggle for survival. Now, if you are a bacteria, apart from developing the ability to produce weapons that can kill others, what else do you want to produce? Don't you want to produce defense to protect yourself from the same kind of attack by others? So the ability to develop antibiotic resistance is also an ability that bacteria have mastered by evolution. It is their battle, it is their territory, it is their strength. The antibiotics that are produced is the strength of bacteria, and they have an equally strong ability to develop defense, antibiotic resistance. Now what do? We found out antibiotics, we figured out that a particular chemical can kill a particular kind of bacteria. We exploited this, we manufactured this as medicines, and we started giving it against this bacteria. It killed. But the amount of drug that is required, the amount of antibiotic that is required to eliminate the entire colony of bacteria is to be given for that duration. Otherwise what will happen? Let's say there are million bacteria that are sensitive to chloramphenicol or penicillin. Let us consider penicillin. You give penicillin, they start getting killed. These bacteria start getting killed. Out of a million, let's say 500,000 is dead. What will be the feeling of illness in you? You have a million bacteria in your body, you are feeling very ill, you are given penicillin, antibiotic, half of them get killed. Do you feel better? Does your health start improving? Do you feel better? You start feeling better. Now suppose you continue to take it, the load comes down to 100,000 bacteria. 100,000 bacteria. 900,000 bacteria are killed. 100,000 bacteria is remaining. You will start feeling very good. And you stop taking the antibiotic now. The level of antibiotic in your blood will start coming down. Now there is a lethal dose. When you are taking the course properly, the concentration of antibiotic was at a lethal dose. Lethal dose means the dose which can kill in the bacteria, in the blood, which killed 900,000 bacteria. But you stopped it. The dose started coming down and it came below lethal dose. Now the bacteria will not get killed because of this dose, but the bacteria will feel ill. They are ill. This triggers the mechanism inside the bacteria to quickly find out ways to deal with this. They have the ability to develop resistance. So they will identify what is this chemical doing to their body, and they will start creating defenses against that. Let's say this chemical, this antibiotic is going and puncturing holes in the cell wall. Puncturing holes in the cell wall and punching holes in the cell wall and killing the bacteria. It is doing it by identifying a particular protein on the cell wall. Now what will the bacteria do? It will develop, so it will do some modification in its genes and change the nature of this protein. Target organ, target changed. Now this antibiotic cannot identify the target. This bacteria became resistant. It will not stop there. What will it do? Now it identifies, so this is what I have to do, I have changed this change happens, and this bacteria, whatever bacteria developed this innovation, it will quickly start passing it on to the other bacteria in the colony. Conjugation. Antibiotic resistance get passed on. You are still infective. You are still infective, but you are feeling better, but you are still infective. Let's say this bacteria which developed the resistance is passed on to a family member of yours. Same kind of symptoms, same kind of course of illness, all of that is happening. So you will feel like, anyways, the same tablets I took, it helped me, I will give the same tablets to my family member also. Now, it doesn't work. Bacteria you can't see, you can't control them. They multiply in, what is that, their multiplication number. They can multiply every half an hour if the conditions are favorable. Within a short period of time, all the existing bacteria of that species can become resistant to penicillin. Now anybody who is getting infected with the resistant bacteria and being treated with penicillin are strengthening the penicillin resistance in those bacteria. And anybody who is getting infected with non-resistant bacteria and getting treated with penicillin, that bacteria is getting eliminated. So this particular species of bacteria over a period of time which survives on the planet will be those who have penicillin resistance. So a majority of them have developed penicillin resistance. You will not be able to treat the bacterial infection of this particular bacteria by penicillin. Over. You see this? So these are all not just simple mechanisms of transferring genetic material, very, very powerful mechanisms which are changing the course of human interaction with these organisms. Our interaction with bacteria are very good, they help us a lot. But our obsession to eliminate them by trying to find out ways, by trying to find out defenses against them, which is playing to their strength. Antibiotics have put us at great risk. Short term, yes, we develop the ability to fight the bacteria, we are eliminating them, all of that is happening. But this is a losing battle that we are fighting. As we move forward, antibiotics will be one of the greatest contributing factors that would lead to human beings getting killed by bacterial infections. We are strengthening our enemies. Not enemies who, it's the interacting strongly in favor of the bacteria. Alright, anyways, leave it. So again, let's not go too much more, any further. Okay, going forward, bacteria again, chrysophytes, dinoflagellates, euglenoids, slime molds. These are all protista, protozoans. Chrysophyta, dinoflagellata, euglenoids, slime molds and protozoans. So in the 5 kingdom classification, you know why each of the organization is appearing in the form of protista. Kingdom protista is like the orphan kingdom. Why? Kingdom of orphans or destitutes. It's a destitute kingdom. All prokaryotes go into Monera. All unicellular eukaryotes go into okay. All prokaryotes go into Monera. Unicellular eukaryotes, forget it. All unicellular or multicellular slime mold forming organisms, saprotrophs, go into fungi. Autotrophic multicellular organisms go into plantae. All non-cell wall containing multicellular organisms, heterotrophic, go into Animalia. Those which don't fit into any of these go into Protista. Protista is like a refugee camp. You identify something, keep it in Protista, we will understand it further. Once you understand it further, you push it into one of the 4 groups. Anyways, alright. So see here, has anybody made syllabus map somewhat similar? No? Not so detailed. So if I give it to you, you will dump your syllabus map and start reading from this. It's okay. But as long as you have done it, it's fine. Okay. See, everything is there. You just, for in Protista, chrysophytes, what do we have? Diatoms, desmids, dinoflagellates, goniolacs, reptiles, euglenoids, euglena, the structure, photosynthesis and nutrition, slime molds, plasmodium stage and fruiting bodies, 2 stages in that. Protozoans, amoeboid, flagellated, ciliated and sporozoans, 4 type. Examples are also given. Structure of fungi. Mycelium hyphae, septa, cenocytic hyphae. What are all this? You will have to know. Reproduction in fungi, vegetative reproduction, asexual reproduction, sexual reproduction, plasmogamy, karyogamy. See all the terms that you have to remember. Asexual reproduction, spore formation, sporangiospores and conidiospores. These are the 2 spores given in NCERT. If you go into some other books, there will be more spores given. But these 2 you should know clearly. Sexual reproduction, plasmogamy, karyogamy. Plasmo is the cytoplasm, karyo is the nucleus, plasmogamy is the fusion. You will have diagrams for liverworts and mosses. When you are making diagrams, liverworts and mosses have diagrams. So specific diagrams for those. You see, one by one as you go, the syllabus map will give you a framework of everything that you have to study. Everything in syllabus map you should know. Even if you spend a few days in just learning and revising those things, that is good to do. Some of you are asking, no, how do I go about learning physics? I am not able to solve problems. Solving problems is very glamorous. It looks like, it feels like you know a lot. But solving problems is not necessarily the one which will help you score high. There are a lot of other things which you can quickly get in, that will give you confidence. That will give you the fundamental understanding and confidence, and as you gain confidence, go on to more higher levels of solving the papers. Don't start doing the complex things in the initial stages, which will bring down your confidence. It's a game of confidence. Exam performance is a game of confidence. Your mind should start believing that you are capable of doing it. To do that, you should incrementally make the mind more and more confident. How do you do it? Go from where you are to where you can be. So you should always keep expanding from the basics. Small incremental growth. Understand? So don't worry about your scores. What is the score that you have got in the past? What is it that you are getting? Stick to the process. Build incrementally, step by step. Okay? Even at this stage, there is gap to cover. There is a lot of gap to cover. You have the right vehicle. You just have to drive it. Drive the vehicle, you will cover the gap. In between, if you start getting doubts whether this vehicle will take me or not, should I take some other vehicle? I am used to a particular vehicle. Let me get onto that vehicle only. Stick to the vehicle. It will take you. And as you keep driving, you will start seeing others in the race. All right. Good thing for you is you don't have anybody else to look at. You just have to drive at your own pace and keep on improving the pace of driving, improving your intensity of effort. Wherever you are at this point of time, try to be a bit better. Keep doing this for the next 252 days. Then we will see how we have driven. We will not do any comparative testing with any other group of students. It means nothing. All of that drama that goes around All India Test Series, compare your rank with others in the country, all of that is good for somebody who gets kicked from positioning themselves against others. It just motivates. Otherwise, just stay calm, do your work. Keep doing your work. No matter what you compare yourself with, it can help you get motivated. It can, for most of the people, it will lead to anxiety. See, think about it. You take an All India Test Series, which some 5 lakh people have taken. It's a different game. All right. The desire for human beings to get validation is the basis of the business of test series. Every day we want somebody to tell us, oh, you are doing good, okay, don't worry, keep going, keep doing. That is the desire for validation. Now that desire for validation is pumped up. Your preparation can be better by competing with others. Rank predictor, we will predict your rank. Will you be happy if your rank predictor tells you 50,000? No. Those who get the rank in the rank predictor in 2 digits, 3 digits, will feel like, oh, I should push a little bit further, I can grow further. If your rank goes into 4 digits, it increases anxiety for you. Anxiety will make you feel worried. More and more worry will make you go down in terms of the efficiency of preparation. Efficiency of preparation goes down, you eventually go down in your performance. Alright. So baseline should not be the need for validation, somebody validating you. The baseline should be absolute trust on your ability. Absolute trust on your ability. That is the baseline. everything is possible for me. It is dependent on my effort. I will continuously work to make my effort better. That's all. So there is zero question or doubt about possibility, absolute possibility for achieving any dream. Second, clear understanding that it is dependent on effort. These two, no need to keep revising it or revisiting it every day. What will make the possibility a reality is how much effort you put in, and I will continuously work to make myself capable of putting more and more effort. That's all. Had you ever done 10 to 12 hours of studies in a day for a week at a stretch? Had you done it in your life ever? 7 to 10 continuous days? Not even 3 days, but you have done it for more than 30 days now. You have done it for about 35 days. So that's a great thing. Keep mastering that. Now the quantity has consolidated, quantity has fixed. Whether you are efficient in that, you feel sleepy, you feel tired, you get the headache, you get irritated, frustrated, all of that is there. But you are sitting for 11.5 hours every day at one place. Stage 1 clear? Your mind will start getting confidence that it is possible to sit at one place. When the body stops creating trouble for you, the intensity of your preparation will increase. My strong, the reality is there is a lot of things to do. We are nowhere in the top level of competition yet I say. Ability wise, no problem. But there are a large number of people who have put in significant amount of time in preparation, in the right direction, with the right resources. All of that has happened. So there are a lot of people who are ahead of us. That's the reality. We have put out a right process. We are all following the right process. Let's run the race with ourselves as the focus. Finish it. Then when the results come, we will see where has our preparation taken us. Correct? My experience, my wisdom of having dealt with these things for a very long time, and my understanding of human mind and performance tells me that if every day, every session, each one of you sticks to the process, because I have interviewed every one of you. I have spoken to every one of you, and I have put my stamp of approval for each one of you to be into this program. So there is nobody here who has not been approved by me to be a part of the program. I know for sure that if you can stick to every session as the challenge and coming out successful for every session, by the end of this duration, each one of you are capable of crossing 600. No matter what you are feeling about your physics or chemistry or ability to solve problems, all of that will get corrected. Right. I was telling somebody here only in this group only, example. How the exponential outcomes will happen for human mind. You have put in 10 years of effort in a particular business. You have put in 10 years of effort in a business. You have done everything right for 10 years. You have reached the stage where your income is 10,000 rupees per month. Whereas somebody who is your friend or colleague, they are all earning 50,000 rupees per month. They are laughing at you. You have put in 10 years of time in the business. After 10 years of working double than what we are working, you are getting 10,000, I am getting 50,000. They will tell you, put in another 10 years of effort, you might reach 20,000. Alright. It is exponential. It is like doubling. Let's say if 2 is multiplied 10 times, it is raised to the power of 10. What is the number? 1024. So 2 raised to 10 is 1024. But if you do it again for another 10 cycles, it is 1024 into 1024. It's a million. Do it for another cycle of 10 times. It's a billion. It's a billion. It's the power of compounding. It's the power of exponential development of human mind. Human mind grows exponentially. You should just stick to the process. Stick to it, stick to it, stick to it. That's all. Alright? If it is not to happen, it will not happen. But if you stick to it, what is good, what is right will happen. Alright? Success is dependent on you. Your ability to sit at one place for 12 hours every day and do committed work for 1 year is massive. And this is what you have, if you continue with this during your graduation also, right out of your graduation you can become a civil servant. You can clear UPSC civil services and get into IAS, IPS, IFS, that's all. So what foundation is being laid is the foundation of tremendous ability, competency development. Those competencies will help you in whatever you want to do. It is not teachers, it is not the books you get, it is not the resources you have, it is not the materials, it is not the support system, it is not the money you have, nothing, it is you. Whether you want to become successful in business, you want to become a civil servant, do IAS, IPS, become a foreign service officer, get into politics, do whichever course you want to do, all of that, success in all of that is dependent on you. My job is to make you realize that. That is what I told you in the orientation also. You are capable. My job through the program is to make you realize that. That's all. All of this exercise is to do that. And because we have a shorter duration of time, the duration is short, I am helping you with the resources also. I will give you the syllabus map, I will make it and give it to you. But after you develop the ability to do it, I will also give you the level 1 notes, I have the perspective notes from my side, but only after you have learned how to do it, after you have done it. All of us will help you with the post-test analysis notes from our side also, but only after you become comfortable doing it. Alright? So, have zero doubt, okay? Your trust on your abilities should be bigger than the trust on God. Because it is God who has given you the ability, no? Trust on what God has given you. He has already given you. What are you asking for more? Alright, you have already been given a bank and you are asking God for 10 rupees to buy kurkure. Alright, you have the bank, go take it. You just have to unlock the password. Not my Wi-Fi password. Alright. Okay, so plantae, algae, bryophytes, pteridophytes, gymnosperm, angiosperm. Angiosperm, again each one, whatever the further division, whatever the further classification given in NCERT. Our reference books expands it further, we will stick to NCERT. Chlorophyceae, Phaeophyceae, Rhodophyceae. Bryophytes further divided into liverworts and mosses. Pteridophytes, ferns and horsetail. These 4 put together will give you a confusing question. Which one is a bryophyte, which one is a pteridophyte? That itself, identifying which one is a bryophyte, which one is a pteridophyte itself becomes a problem. Alright? Gymnosperms, Pinus and Cycas, Angiosperms, Monocots, Dicots. That's all we will be studying. Kingdom Animalia. Overview of the animal kingdom, classification criteria in animal kingdom. It is all animals are multicellular eukaryotic. The levels of classification criteria are levels of organization, symmetry, body plan, coelom and segmentation. Body plan, coelom and segmentation. We can further divide it. Alright. Now you have these, no? Level of organization, symmetry, body plan, coelom and segmentation. You have 5 points. 5 points. 5 points become each column. The 10 different phylum of animal kingdom, the 10 different phyla of animal kingdom will be the first column. So for each of the phyla, what is the level of organization, what is the symmetry, body plan, coelom, segmentation. You make one table for that. That table will be a complete summary or comparison between all the 10 phylum of animal kingdom. Alright, that's how you will expand it into level 1 notes. Here you are just looking at the five parameters of classifying animal kingdom: levels of organization, symmetry, body plan, coelom and segmentation. To make level 1, the same thing will expand into a comparative table. Then capsid, structure of, that is over, animal kingdom classification overview over. Why this one? Because this is the second chapter, biological classification. We are not going into detailed discussion of animal kingdom or plant kingdom yet. Viruses, structure of virus. So what does the virus have? Three parts of the virus: capsid, envelope, genetic material. Capsid, envelope, genetic material. Again, from outside to inside, from inside to outside, what type of genetic material will be there? What is the capsid made up of? These are the questions. Alright. Then types of viruses. Bacteriophage. In the bacteriophage, you will have to study the T4 phage structure. Plant viruses. TMV is the reference virus that we will be studying, tobacco mosaic virus. Animal viruses, influenza virus and HIV are the ones we are studying. Reproduction in viruses, I told you, lytic cycle and lysogenic cycle. Lytic cycle, attachment, penetration, biosynthesis, maturation, lysis. Over, no? A bacteriophage will sit on the bacteria, attachment. It will push its genetic material into the bacteria, penetration. Biosynthesis, genetic material and protein, both of them will multiply within the bacterium. And maturation is assembly. Lysis is the host will be destroyed and removed. Okay. I was telling you the life lessons from those three: conjugation, transformation and transduction. Conjugation from trusted people. You need to have a set of people whom you can trust. In conjugation, there is exchange of genetic material, no? So trusted people with whom you can share things which you cannot share with others, and you are absolutely sure of that not harming you. So who are those people? It will be your family and close friends. You should not keep sharing everything with everybody. Sharing of things that are personal to you should be only with people whom you can trust, who do not judge you. Sharing of your problems should be with people who are non-judgmental. Alright? Sharing of personal matters is only with people with absolute trust. It cannot be every acquaintance in the class. It cannot be everybody in your friends group. You should be having that extreme level of trust. That is conjugation, level 1. Transformation is in a public setting. This is a classroom. There are 48 people here. This is the cream of Meghalaya. So you will only make good friends here in this group. But when you are outside, there are all types of people. What you take determines what happens to you. Whether it is a thought, whether it is a friendship, whether it is the work that you share and do, what you take, you should be careful. Transformation will tell you the influence of the surroundings on individuals, your life, how your life gets changed because of the surroundings. You should be very careful about what you will let into you through any of the sense organs, what you see, what you hear, what you speak. So, good acquaintances, good sources of information and knowledge. Transduction is, if you get in contact with people who are forceful, but who have a negative agenda, you will be destroyed. You will be destroyed. So let's say somebody has come, a terrorist has infiltrated into a particular region. A terrorist has infiltrated into a particular region. One person has come in. One person who is a terrorist, who has the intention of killing people, who has come into a particular region. As soon as he comes, will he start killing? As soon as he comes, will he start killing? He might, like if he was like Kasab, 2008 Bombay attacks. They had come with the intention of killing right away. They entered, they kill. As many as possible, done, over. That is lytic life cycle of viruses. Lytic life cycle. Lytic life cycle, just enter, just spread out and kill. Lysogenic life cycles are like sleeper cells. Sleeper cells of terrorist organizations. They will be individuals who will be with the community, who will be with you, who will be around you, who will be in the same community. They will be taking opportunities to poison the minds of somebody else, increase their numbers to wait for the critical mass to develop. When the mass of people is critically done, and when they see the conditions becoming favorable to mount an attack, that is when they go into the lytic cycle. They will start attacking. So, conjugation, transformation, transduction. Have a good number of people whom you can trust, where conjugation is possible, soul to soul connection. Be careful about what you take from the surroundings, and be cautious about people who can function like viruses. Your ability to identify viruses in the society is very important to keep the society healthy. Viruses are the ones which will destroy. You understand? See? Biological classification chapter is telling us how we should build our social life. Okay. Then, viroids. What is a viroid? And another bit of expansion would be virus, viroid, prions. Can you tell me the difference between virus, viroid and prions? Protein coat genetic material. Virus, they have a covering around their genetic material, virus. Plain genetic material going and incorporating into host DNA or RNA is viroid. Prions, what are prions? Do you remember a disease? That sheep. What sheep is that? I forgot. They go and rub their prion disease. Mad cow disease. Mad cow disease is the disease caused by prions. Prions are infective proteins. Infective proteins, they will go and damage the nervous system. Mad cow disease. Alright. So, these are the genetic material. Lichens. See, viroids have circular RNA, lack of protein coat. Just your syllabus map will give you basic information. That's all. If something else comes beyond this, we will see later. But at least this much we know. Something about everything is a very powerful preparation strategy. Something about everything is the baseline that we have to achieve quickly. Syllabus map and level 1 should take you there. Lichens, structure and types of lichens. Crustose, foliose, fruticose. This has come as question. Crustose, foliose and fruticose. Crustose is like a crust, enlarged. Foliose is like leaf-like lichens. Fruticose is like a fruiting body. Role of lichens as bioindicators, pollution indicators and symbiotic relationships. When you go in for your level 1 notes, you should identify who are the pollution indicators. What lichens will act as indicators of what pollution, and what are the symbiotic relationships that the algae and fungus as a combination are having in the nature. That's all. Going one step further. Okay. So are you able to understand level syllabus map leading on to level 1? And once you are done with this, you have the basic revision material ready. Then you take up the perspective based notes. So you have about 5 perspectives for each subject, one small booklet or one small book for your syllabus map, a little bigger book for your level 1 notes, one book each for every perspective. 5 plus 2. Around 7 to 8 books is your linear revision material, linear revision notes. That's all. Post October, see till October you are having this syllabus map is over. Level 1 you are doing, perspective based notes you have to do. Till end of October you are doing that. After October, your testing frequency will become one test every 4 days. One test every 4 days. October, November, December, two months. January, February, March, your testing frequency will become once every 3 days. Once every 3 days. One day before the test. One day before the test and on the day of test. So it is 1.5 days before every test. You will revise your entire post-test analysis notes from the beginning. So every time before the test, you will revise your post-test analysis notes. Do that for coming Monday test also. Every test, one day before the test, on the day of test, you will try to revise your entire post-test analysis notes and take the paper. Every day for about 2 hours. Every day for about 2 hours, you should revise your linear notes in a sequence. You should mark the books. Let's say you mark it as 1, 2, 3, 4, 5, 6, 7, 8. 1A, 2A, 3A, like that for physics, chemistry, biology. Or you can mark it as B1, 2, 3, 4, B1, B2, B3, B4, C1, C2, C3, C4, P1, P3, P goes on like that. So you will complete one of all the books, all the subjects in a sequence. Then you will take 2, all the books in a sequence. 3, book number 3, all the books in a sequence. Book number 4, all the books in a sequence. But you are doing only 2 hours. 2 hours of linear notes revision post October. Now you are making the linear notes. You are making the linear notes. A significant amount of time is going in writing. So 2 hours of linear notes revision. Rest of the time, your 3 hours you will be in a session. 5 hours done. 6 hours you will be working on post-test analysis notes every day. 1.5 days before the test that you take, revision of post-test analysis notes. You understand? If you need about, let's say, 20 hours to complete revision or 10 to 12 hours to complete revision of each subject, for all the 3 subjects, you will need about 30 hours for revising linear notes. Once every 15 days, you are completing revision of all the chapters of all the subjects from your linear notes. So you are ensuring that nothing of the basics are missed out. Everything about NCERT you are revising repeatedly. So from October, by October if you are completing November, December, January, February, March, 5 months, every 15 days is one cycle of revision. You are not writing, no, you are revising, reading from your notes every 15 days. You would have done 10 revisions of NCERT from your notes. 10 revisions. And post-test analysis will always be lagging. You will try to cover off the notes making. As you keep moving forward, the notes will become faster and you will be able to catch up. By end of December at least, if you are catching up with everything, if you become up to date with the post-test analysis notes at least by the end of December, January, February, March, lot of revision of the notes, and 1.5 to 2 days would be enough for you to add on to the notes to whatever you already have done on a once in a 3 days test you are doing. Get it? The entire month of April, revision of all these linear notes, 30 hours you require. Once you do 10 times, you will be able to complete revision of all the 3 subjects in half time, 15 to 20 hours. 20 hours for completing one cycle of revision on the linear notes. That is taking care of 70% of marks on NEET. And all the post-test analysis notes that you have done. That will take time. That quickly run through. You would have run through that so many times before every test. So you just look at it, you will know it. If this much is not enough for you to cross 600, nothing is. Alright, you will cross. Chlorophyll A, chlorophyll B and phycoerythrin. Chlorophyll A, chlorophyll C, phycoerythrin. Chlorophyll A, chlorophyll D, phycoerythrin. Now it comes down to whether you know that red algae has chlorophyll D pigment. That's all. Easier ones will be if one of the options will have fucoxanthin. You know it doesn't have, so eliminate fucoxanthin. If one of them does not have chlorophyll A, if you at least know that all the three types of algae have chlorophyll A, then that also can be eliminated. So know that chlorophyll A is present in all the three types of algae. Fucoxanthin is what gives brown color to brown algae. Phycoerythrin gives red color to red algae. This much if we know, even if the confusion between B, C, D should not be, because green, brown, red, if you remember in the same sequence, B, C, D in the same sequence. As you repeatedly revise, you will fix it. It will get fixed in the mind automatically. Then reproduction, vegetative, fragmentation, asexual spores are produced, zoospores and similar kind of spores. Sexual reproduction, isogamy, anisogamy, oogamy. What is the difference? Iso means similar, aniso is dissimilar, oogamy means ovum is bigger. Ovum is bigger. So is oogamy also a type of anisogamy? Is oogamy also a type of anisogamy? It is. Oogamy is also anisogamy. Alright. Bryophytes, general characteristics, thallus-like or erect plant body found in moist shaded areas and soil. Classification of bryophytes, liverworts, mosses. Liverworts, example Marchantia, mosses, Funaria sphagnum. This will come. I told you some 3 or 4 will be given and you will be asked to pick the odd one out. There can be other 100 types of bryophytes, but this you can't forget. You should not forget what examples are given in NCERT. So in your syllabus map, in what I will give you, there will be no example that is outside of NCERT. Because I don't like to remember examples, and also fortunately, exam also doesn't give you too many questions with examples outside of NCERT. It suits us. People like me also can do well on exams like NEET. Alright. If it is absolutely memory based, problem. I will not take that exam. Mosses, Funaria, sphagnum. In the ocean of examples that we get in so many things that we study, isn't it comforting to just limit ourselves to NCERT examples? And when we know these are the only examples that we have to study, mind will also start feeling like, okay, let's do it. When you open a book and there are some 10 categories of things and each category has 20 examples, the moment you see it, mind will just come outside of your body and look at you. Do you want to do it? I think we should not do it. Alright, just go. Again, movies are great sources of learning. Have you seen Lage Raho Munna Bhai? No. You watched? He is supposed to go and give a lecture on Gandhiji to impress the heroine. But he knows nothing about Gandhiji. He only knows that dry day is there on his birthday. Alright. So he goes to the library, Gandhian library. The entire library is filled with books written by Gandhi and about Gandhi. He goes and picks up the smallest book. In an entire library of books, he goes and picks up the smallest book available on the topic. That's the right way to study. When you don't know anything about something, you should try to pick up the most basic material about that thing to start with. Then next little bit more. If you directly open the biggest book, you are gone. Bryophytes, reproduction. This is also important. What is dominant? Is it the gametophyte stage or the sporophyte stage? Life cycle based questions also are common in plant kingdom. In bryophytes, gametophyte stage is the dominant stage. Sporophyte is attached to the gametophyte. Pteridophytes, general characteristics, they are the first vascular plants. They have true roots, stem and leaves. Classification, Coelopsida, example Coelotum, Lycopsida, Selaginella, Lycopodium. Selaginella is important as a protein source, food source. That will come as an example for food also. Okay. Spinopsida, Equisetum. Equisetum is referred to as horsetail. Alright. Pteropsida, ferns. Reproduction, sporophyte is dominant, requires water for fertilization. You see between bryophytes and pteridophytes, in bryophyta, gametophyte was dominant and sporophyte was dependent on the gametophyte. Here sporophyte is dominant. Obviously the gametophyte will be attached to the sporophyte only. Sporophyte is the, in a regular tree if you see, the entire plant body is like the sporophyte. The flower, the reproductive structures is the gametophytic stage. Alright. Gymnosperms, general characteristics, naked seeds, no ovary wall, seeds are exposed. Classification, cycas, unbranched stem, pinnate leaves. Pinus, branched stem, needle-like leaves. Ginkgo, fan-shaped leaves. Reproduction, heterosporous. Heterosporous, microspores and megaspores are produced. Angiosperms, general characteristics, they are flowering plants, seeds enclosed within fruits. Classification, dicotyledons and monocotyledons. Dicotyledons, 2 seed leaves, reticulate venation. Excuse me. Monocotyledon, single seed leaf, parallel venation. When you go in for level 1 notes, classification should translate into table of comparison of all the different categories. Reproduction. In reproduction, pollination is a characteristic feature. Pollination by insect pollinated, wind pollinated, variety of pollination methods. So when you go for level 1 notes, you will just elaborate a little bit on pollination. What is it and what are the different medium through which pollination can happen. See, 3 chapters over. I am going a little with explanation also. If you have done it repeatedly, you will just browse, you will just go through. What you don't know, your mind will tell you, this is not known. Go to the page number, pick it up, come back, done, over. Animal kingdom, our favorite topic, no? Right. I did not like reading this, neither did I like teaching this for a long time. Okay. But I had to figure out ways of understanding it and teach it. Even now, I don't like to remember the examples, so with examples, it's a conscious choice to not remember the examples. I don't want to put in so much of my mental energy which I value. I value my mental energy, mind space. I don't give so much mind space of mine to remember examples. But concepts are, we can understand. There are good ways of understanding animal kingdom which gives us a fair idea about the life around us. If you just eliminate examples from the equation, animal kingdom chapter becomes interesting. Alright? But our mind goes to the difficult ones. Right? If there are 3-4 children, 2 children are requesting for food. One child is refusing food and running away. The mother will go behind the child which is refusing food and trying to convince the child to eat. The child who is asking for food is told, hey, wait, you wait please. He's not eating, I'll make him eat first. Alright. So it's a tendency to break the most difficult challenge. That is the tendency which will make you waste time on problems which will take more time. Correct? We get a kind of kick from that. When you solve complex appearing problems, your mind will get an ego boost. For that ego boost, we will waste time in exam on those problems which we at the beginning of it only knew that we might not get an answer. So, don't try to prove to yourself that you can do complex things. Be strategic in the way you approach your papers. Basis of classification. What is the first basis that we saw? In the biological classification also different basis for classification. Levels of organization. Cellular level, no tissue formation. Tissue level, group of cells. Organ, tissue groups together. Organ system, organs working together. That's all. Here, when you go to level 1, for all of this, these parameters, the basis of classification should be the first, there should be different columns. All the phylum should be in the first column, each row. The logic is simple. How much the paper can accommodate. If there are more, whatever is more should be in rows, so that the span of the table doesn't go too wide. It goes long. Assuming that we are writing in a portrait format. We are writing in a portrait format on the paper. We can extend below, downwards. If we go too wide, it will look clumsy. Symmetry, asymmetry, there is no symmetry. Radial symmetry, body parts arranged around central axis. Bilateral symmetry, body divided into 2 equal halves. What kind of symmetry do we have? What kind of symmetry do we have? Bilateral symmetry. If it was radial symmetry, okay. What kind of symmetry does a banana fruit has? One banana. Banana. Radial, not radial, bilateral. You can cut it into 2 equal halves in any one of the 2 planes. Vertically you can split. If you cut it in any longitudinal plane, will it be the same? May not be. May not be. A properly shaped apple might be radial symmetry. You cut it in any way, it will still be the same. It will appear the same. Alright. Chili. Chili. Maybe bilateral symmetry. Depends on what chili you use. Diploblastic and triploblastic organization. Here some more concepts come which will help us understand other animals also. Diploblastic is 2 germ layers, ectoderm, endoderm. Triploblastic is 3 germ layers, ectoderm, endoderm and mesoderm. When you go further, there should be one table which will give you comparison of the structures that come from each of these 3 germ layers. What will come from ectoderm derivatives, mesoderm derivatives, endoderm derivatives. Those 3 should be there in one table. That is also a question. That is also a question that appears repeatedly. Coelom. Coelom is the body cavity. Acoelomate, pseudocoelomate, and coelomate. Acoelomate is no body cavity. Pseudocoelomate is body cavity not fully lined with mesoderm. Coelomate is body cavity lined with mesoderm. It is the modification of the coelom that we find, or the body cavity that we find, which is around all our internal organs. Our pleural membrane is the modification of coelom. Renal capsule is the modification of coelom. The capsule around the liver, hepatic capsule, Lissen's capsule, the coverings we have around, omentum that we have around the intestines, all of these are modifications of the body cavity or the coelom. Imagine it this way. This room is the body during development. This room is the developing embryo body. A big balloon is the coelom inside this room. A big balloon is the coelom inside this room. Fluid filled. Fluid filled. Each one of us, let us assume, are different organs. Each one of us are different organs. Is it possible for us to get ourselves surrounded with the balloon but not go inside the balloon? Is it possible? Is it possible to surround yourself with the balloon but not go inside the balloon? Not possible? I'll give you a situation easy to imagine. I'll give you a balloon and I'll give you 4 tennis balls. I'll give you 4 tennis balls, a big balloon and 4 tennis balls. You have to push the tennis ball into the balloon in such a way that the tennis balls are not inside the balloon. Inside the balloon there is still air. You have blown the balloon. You have to make sure that the balloon surrounds the tennis ball completely, but the tennis ball should not get inside the balloon. It should not be inside the balloon. It should be surrounded by the balloon. Possible? Can you do it? Can you do it or not? No? How will you do it? This is the balloon. I take the tennis ball. I will push the tennis ball inside. So now it's here. I will hold it, take out my hand and tie it. Possible? Can I do that? Now the ball is surrounded by balloon, but the ball is not inside the balloon. I'll do it with all the balls. I'll push all the balls into the balloon like this. Later what I will do, whatever is not surrounding the ball, whatever balloon is not surrounding the ball, those balloons I will fuse to each other and make it into sheets. I'll stick those balloons to each other, the two part, whatever balloon is in contact with each other, I'll stick the balloons and make it into sheets. Now would it be like a sheet with balls inside the sheet surrounded by balloon, surrounded by this sheet? Is it possible? You're not able to visualize. I should have brought balloon. I did not think that I'll elaborate this. This is now balloon has become flat like this. Wherever the ball is there, becomes like this. Can we make the balloon like this? Inside there is ball. Possible? Is it possible? Is it possible to do it like this using the balloon? Yes. You have pushed the ball, three balls inside, and wherever there is no ball, throughout here, now every ball is surrounded by two layers of balloon. Every ball is surrounded by two layers of balloon. Between the two layers, what is there? Between the two layers of balloon surrounding the ball, what is there? Air. Now imagine there is fluid. Imagine there is fluid. Okay? Here, I will just make this hard. I will make it hard. So I will just block this by putting some hard substance into it. Now do I have three isolated compartments? Now do I have three isolated compartments? They all came from one big balloon into which these things got pushed, balloon surrounded it, and whatever was in between got sealed. Isolated compartments. Now the covering of this, I will make it the outer one hard, the inner one thin, and I will add fluid into this. Pleura. Alright? This one, I will make it fibrous. And hard. That would become renal capsule around the kidney. Somewhere like this, one more is there. I will make the surrounding also fibrous and hard. What is this surrounding? Liver, Glison's capsule. Are you understanding how the coelom or the body cavity develops around all of these organs? When you look at how this development is happening, it's like this. It's the same empty cavity that was there inside. When you look at the embryonic development, when we get a question related to embryonic development, I'll explore that. As the development starts happening, when you go back today evening and see the diagrams related to embryonic development in human reproduction, development of the embryo. There is a lot of fluid filled cavities inside. There are cells, there are some outer cells, inner cells, fluid forms, and suddenly you will start seeing what is that movement? What do you call the movement of cells? All the germ layer formation. So suddenly the endoderm forms, ectoderm forms, mesoderm forms, and suddenly they will tell you these are the structures that came from this. Intervening structures, when you see how each one transforms, I'll try to get some videos to show you that whenever those questions come. So it's nice that way. You understand? Coelom. What is coelom? Coelom, or the body cavity development, you see here, no body cavity, acoelomate. You look at the level of organization of these people, of these organisms, which do not have a body cavity. You look at the level of organization of those which have pseudo cavity or pseudocoelom, and then look at the level of organization of those organisms which have a coelom, body cavity. So the complexity of development and organization is correlating with the level of maturation of coelom. Those which do not have coelom have a very simple body structure, simple functioning. They are not complex. So coelom created possibility for the organs in the body to become specialized. Specialization requires isolation. If you are not isolated, there will be generalization. Everything will be the same. Isolation is necessary for specialization. Alright. Why I proposed a fully residential, no phone, no internet campus? Specialization, transformation. Without a cocoon, the caterpillar does not become a butterfly. Alright? So it first builds the energy, it eats and becomes and grows. Growth is complete outside. When it is ready to develop, evolve, it isolates itself. Observe nature. Observe nature. Every evolutionary specialization in any organism is happening after isolation. There is isolation. Correct? Embryo development. Does it happen in open waters? Even those organisms which are having external development, even in that, there is some isolation brought about by a membrane or something which will cut it off from the outside world. The egg shell is there to allow the embryo to mature and evolve. Transformation requires isolation. Alright. All those people who are thinking that you have been sent to Ukraine war, your friends or relatives who might be those who can't get in touch with you, they are the ones who will tell you how you have changed. Your parents are still meeting you once in 15 days. The way you talk, the way you will walk, the way you will be, everything will be changed. Alright, anyways, coelom. So we have given you a coelom. Outside you are all acoelomates. Going around, talking to everybody, just being with everybody. During exams, you become pseudocoelomates, achieving some level of success. But if you are in Star 30 program, you become eucoelomate or true coelom, achieving transformation. Alright? So, coelom is very important. Segmentation, metameric segmentation, body divided into segments. Metameric segmentation is true segmentation, what you can find in some organisms where any segment of the body is same. Of the body is same. Do we also have segmentation? We do have segmentation, but it is not metameric segmentation. Our vertebral column, segmentation. Rib cage, segmentation. We have segmentation partially. Not true segmentation or metameric segmentation. Notochord. Chordates have presence of notochord. Non-chordates do not have notochord. They have absence of notochord. Again, notochord is also one of the advancements. Notochord is also one of the advancements which allowed for the development of nervous system. Nervous system maturation happened well because of the formation of notochord. It gave the axis for the development of the vertebral column. Anterior-posterior axis was fixed because of the notochord. It provided the axis for the development of vertebral column and the spinal cord. Alright. So which allowed for multi-level maturation or multi-level specialization of the nervous system. Alright. Somebody wanted to do business, no? One thing, if you are allowed to do, I will do business. Who is that? You. Okay. Learn from this. See, the more problems you solve, or the problems of a bigger number of people you solve, the bigger business you will make. Alright. As you come higher and higher in the level of organization of animals, you see that different types of problems of survival are tackled in more and more specialized ways. So there is some solution for every problem. There is some solution for every problem. That is why higher chordates are most successful life forms on the planet. So if you want to make a lot of money doing business, you have to find a problem worth solving. Alright, if the problem that you solve or the solution that you provide is relevant for a large number of people, then you have the chance of making a lot of money. You cannot start with the intention of making a lot of money. You should start with the intention of identifying a problem to solve. And a problem to solve, which a solution which people will pay for. First step is identify the problem. Second step is experiment with solutions. Third step is try if people like it or not. Fourth step is ask money for that. See the willingness of people to pay for the solution. Then fifth step is start selling it in small numbers. Then learn from it, scale it up, make loads of money, pay me later for giving you the initial ideas. Alright? Okay. There is a lot of payment that you have to keep making for the rest of your lives. Oh, camera is running, no? Okay. Then people who watch this will start thinking that this fellow is asking for money otherwise from students, collecting money. Classification of animals, non-chordates and chordates. Most of them are non-chordates, just porifera, pore-bearing animals, mostly marine, Coelenterata or Cnidaria, aquatic, radial symmetry, tentacles, Ctenophora, comb jellies, Platyhelminthes, flatworms, bilateral symmetry, Ashhelminthes, roundworms, cylindrical, Annelida, segmented worms. These are the true symmetry ones. These are the true symmetry, segmentation, metameric segmentation, annelids. Arthropoda, jointed appendages, exoskeleton. Mollusca, soft body, usually with shell. Echinodermata, spiny skin, radial symmetry. Hemichordata, marine worm-like. Chordates, among the chordates, the not so successful but existing, not so prominent, cannot be put into non-chordata because they have notochord, cannot be put into vertebrata also because they don't have the vertebral column development. They are like intermediate forms, Urochordata and Cephalochordata. Urochordata, notochord in the larval tail, Cephalochordata, notochord extends from head to tail. Of the roots. This is important. Diagram will be given and you will be asked to identify the regions. And different options will have combinations, mix and match of these regions. Going from the outside to inside, you have root cap, region of meristematic activity, region of elongation and region of maturation. Which of these four, let's say 1, 2, 3, 4, which of these four will have cells which are yet to differentiate? Region of meristematic activity. These are also non-meristematic cells. Region of elongation is non-meristematic cells. Then why do we call it region of elongation? The cells are being added by these fellows. This is getting pushed. So that appears like it is elongating, but not because of its multiplication, because of the multiplication of the cells in the meristematic zone. Then these fellows continue to become functional, mature. Alright. So root cap protects the root apex, region of meristematic activity for cell division, region of elongation for growth, region of maturation where root hairs develop. Stem characteristics. What is special in stem that we will see? Nodes and internodes. Nodes and internodes. Stem is the ascending part of the plant, buds green in young stage. They support the plant, develops from plumule, supports the growing stem, support the branches, leaves, flowers. Modifications of stem: underground, aerial, subaerial. In each of these there are a few types that are given in your book. Underground, rhizome, corm, tubers. One example for each. Ginger is a rhizome, colocasia is a corm, potato is a tuber. If you get some other examples, cross check with this. Is it like potato? Is it like ginger? Is it like this? That goes on. Aerial stems, tendrils, thorns, bougainvillea. Subaerial stems, runners, stolons. Strawberry is a runner. Structure of the leaf. What all do we see in a leaf? Base, petiole, lamina. Three parts of the leaf. Base, petiole, lamina. Leaf base attaches to the stem. Petiole is what that supports the blade. What you can see as the stalk of the leaf. Blade is the lamina is the green expanded part. That is also called the blade of the leaf. Alright. Venation, reticulate and parallel. Reticulate venation is found in dicot stem, dicot leaves. Parallel venation is found in monocot leaves. All of these have come as questions. All of these have come as questions. If it is appearing very simple, this is all very easy, very good. Easy things only will come in exams. Alright. Easy things come in exams. So those who focus on difficult things, ignoring the easy things, they will do mistakes on the easy things. Overall score goes down. We will not do that. Modifications. Leaf tendrils, spines, phylloid. Types of inflorescence now. Racemose, cymose. Racemose inflorescence, cymose inflorescence. This morphology of the flowering plant will have morphology of flowering plants will have a lot of diagrams in your perspective notes. Your diagram based perspective notes will have a lot of entries from this. You should write simple, easy to understand diagrams, easy to differentiate diagrams in this chapter. You will have to spend some time in making good diagrams. Parts of the flower: calyx, corolla, androecium, gynoecium. Calyx is sepals, corolla is petals, androecium is stamen, gynoecium is pistil, another name carpel or pistil. Okay. This stamen has filament and anther. Pistil has stigma, style, ovary. Fruit. Types of fruit: simple fruit, aggregate fruit, multiple fruit. Simple fruit mango, aggregate fruit raspberry, multiple fruit pineapple. So where are these coming from? Fruits come from flowers. Flowers are a part of inflorescence. So you need to have tabular arrangement of inflorescence to flowers to fruit. Correlation. What kind of inflorescence do the simple fruits come from? What will be the corresponding inflorescence in a plant which is producing complex fruit or the multiple fruit? Alright. How would the inflorescence of strawberry be? From here only, don't go beyond that. The example of raspberry is given here as an aggregate fruit. Try to find out what is the inflorescence in raspberry. Pineapple is given as an example of multiple fruit. Find out how will the inflorescence of pineapple be. It's all leading from one to the other, no? Inflorescence, flower, fruit. Correlate one to the other. Of the same plants which are given as examples. Then it will be, see, they have not given you what inflorescence raspberry has, but they have given you a raspberry as an example for aggregate fruit. Your job to do as a person who studies from NCERT should be to go backwards and find out what is the inflorescence of raspberry. Alright. So a question which asks you the inflorescence of raspberry is also can be considered as coming from the basis of NCERT. You cannot say it is not given in NCERT. So what is given and what is extrapolated, both are to be considered as part of based on NCERT. Seed, dicot seed, monocot seed structure. Alright. So what all you find in a seed? You find seed coats, the endosperm and the embryo. Not the endosperm, the endosperm, cotyledons and the embryo. Anatomy of flowering plants. Tissue system, epidermal tissue system. What do we have in the epidermal tissue system? Epidermis, stomata, trichomes. Epidermis is the outermost layer of the plant body, usually single layered. Stomata, structures in epidermis of leaves regulate transpiration and gas exchange. Trichomes, hair-like extensions, epidermal hairs on the stem, prevent water loss. So you have epidermal tissue, ground tissue and vascular tissue. Epidermal tissue, ground tissue, vascular tissue. You can use the same thing to write chapter by chapter flow charts also. Syllabus map is the tabulation of a flow chart. Still flow chart also, no? Tissue system in plants, epidermal tissue, ground tissue and vascular tissue. In epidermal tissue you have epidermis, and in ground tissue you will have hypodermis, hypodermis, cortex and endodermis. Goes on like that, pericycle up to pericycle. Then vascular tissue will be xylem and phloem. So ground tissue system, based on the type of cells, you have parenchyma, collenchyma, sclerenchyma. Parenchyma, collenchyma, sclerenchyma. These are all simple permanent tissue. Xylem, vascular tissue systems, xylem and phloem. Xylem conducts water and minerals, phloem conducts transports food. Anatomy of dicot and monocot roots. Just see this. Dicot root, epidermis is not given. What is given? Epiblema. So what is epidermis in a stem? We refer to it as epiblema in the root. Epiblema, outermost layer of the root with root hairs. Cortex, layer below epiblema made up of parenchyma cells. Endodermis, innermost layer of the cortex, has Casparian strips. Casparian strips do you find in stem? Casparian strips are characteristic of root. Do you find in monocot roots? Do you find Casparian strips in monocot roots? Monocot root vascular bundle and dicot root vascular bundle. How are they arranged? All of these are the questions that you have to explore. Casparian strips are present in, I will not go beyond anything that is given in this now. Alright, so just by looking at it you will get to know the differences. Endodermis has Casparian strips here. Pericycle layer just inside endodermis gives rise to lateral roots. There was a question that we explored. Endodermis gives rise, pericycle gives rise to lateral roots. Vascular bundles. The nature of arrangement of vascular bundles. Radial, xylem and phloem arranged in different radii. Radial vascular bundles. In stem, what kind of vascular bundles do you find? Conjoint vascular bundles, where xylem and phloem are arranged in the same radius. Here, xylem, phloem. In stem, it is xylem, phloem is one bundle on the same radius. Alright. Dicot stem. You have epidermis, outermost protective layer, hypodermis made up of collenchymatous cells, cortex below hypodermis made up of parenchyma, endodermis, innermost layer of cortex surrounding vascular bundles. You don't find pericycle here. Vascular bundles are present, which are conjoined, collateral, open and endarc. What is an open vascular bundle? What is a what is an endarc vascular bundle? Those things you have to know. Endarc is a reference to what? Endarc was reference to what?
Near the pit.
Near the pith. Is it a reference to the presence of cambium? Open is reference to cambium. What is endarc and exarch? It is with respect to the protoxylem and metaxylem. What is facing inwards in endarc? Protoxylem facing inwards, that is facing the pith, is endarc. If it is the reverse, it is the, see, endarc and exarch is a reference to the orientation of the protoxylem either to inside or to the outside. What is it facing? So those concepts you have to be clear. Pith, central part made of parenchyma cells. Monocot root. Casparian strip is present in monocot root also. So Casparian strip is a characteristic of root. There is no Casparian strip in stem. Casparian strip is a characteristic of root. Pericycle. Pericycle. Do we find in stem? Pericycle. Pericycle is also found in roots. Alright. Epidermis, cortex, endodermis, pericycle, vascular bundle of monocot root is scattered. More numerous and scattered. In dicot root is, vascular bundles in dicot root is radially arranged. It is radial vascular bundles in dicot root, scattered vascular bundles in monocot root. Even in monocot stem, the vascular bundles are scattered. So dicot root, dicotyledons have vascular bundles properly arranged, whether it is stem or root. Monocotyledonous plants are those in which vascular bundles are scattered. Hypodermis of monocot stem is made up of sclerenchymatous cells. Hypodermis of stem is made up of sclerenchymatous cells. This is what will make them tough, hard. Grass. When you pull the grass by your hand, if you are not careful, it can cut your hand. A blade of grass can function like a blade cutting your skin. Why? Sclerenchyma. Ground tissue is not differentiated into cortex and pith. Vascular bundles again scattered, surrounded by sclerenchymatous bundle sheath. Sclerenchymatous bundle sheath is present in dicot when monocot stem. So a simple tabulation of only the frame blueprint gives you so much of information. Questions like secondary growth. And if you notice, all of the things that we have seen in the syllabus so far, all of them have been questions at one or the other point of time. Everything. Isn't it? So everything has been questions. So a syllabus map for you would be a material which is a very solid high conversion material. High conversion material means the number of questions that can come from this will be much more than the number of questions which will come from any individual source of reference that you will study. And this will be the shortest material that you will have. Alright. Again, business. If you are doing a business, see, I am giving some business advice, she is sleeping. How will you do business like this? If you want to do business, you should not sleep. There is something called Pareto principle. P-A-R-E-T-T-O, Pareto principle, 80-20 principle. You know that? 80% of your success comes from 20% of your work. 80% of the wealth is held by 20% of the people. This you can relate to. 80% of the wealth in any part of the world will be held by 20% of the people. 20% of the wealth will be distributed among 80% of the people. It is natural. 80% of your success in exam will come from 20% of your material. This is a part of that 20%. Alright. All the notes that you are making, no? All the notes, your linear notes that you are making, syllabus map, level 1 notes and the perspective based notes. I am making a claim today. I am making a claim to verify after May 4, 2025. 80% of your questions will be, 80% of your marks will come from that linear notes that you will make. Syllabus map, level 1 notes. If you do this properly, you will get 80% of marks from that. I am making claim on record. Alright. Okay. For the rest of the competition is in that rest of the 20%. If you can ensure that you get this 80%, 8 sevens are 56. 560 you will cross with this much, what you are preparing. It is to cross beyond that level, the perspective, the post-test analysis and all will give you the format of doing solving questions. So it is giving you the practice of how to go about solving the questions, but on what basis you will solve questions is these notes. 560 plus more. Secondary growth, vascular cambium and cork cambium. Vascular cambium, formation of cambium ring, responsible for secondary growth. Secondary xylem and phloem is produced from that, produced by vascular cambium. Cork cambium, formation of cork and secondary cortex. Cork is phloem, secondary cortex is phelloderm. Cork cambium is phellogen. Phloem, phellogen, phelloderm. All three put together is bark. All three put together is bark or the periderm. Bark is periderm, no? Periderm is bark. Cork is phloem. Cork cambium is phellogen. Secondary cortex is phelloderm. Alright. So if you are writing it as periderm, you will write phloem, phellogen, phelloderm. If you are writing it as bark, you will write cork, cork cambium and secondary cortex. Alright. So this is also done. 6 chapters, 1 session, revision. Alright. We will be able to do all 32 chapters in 1 session as time progresses. As time progresses. Quickly, tuck, 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 tuck. You should also be in line with that pace, no? Now I have to wait for all of you to respond in a way that I get confidence that you have understood. Otherwise, all of us can have our reference material. You can have these syllabus maps in your book. I can have it on my laptop. I will sit here. You will just look at it one by one. Tuck, 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 tuck. I can go on telling. You will just go on connecting it with what you already have learnt, and we will finish revision of 32 chapters in 1 session. Maybe 4 or 5 hours. Even if it is 5-hour session, we should be able to do that in one stretch as we go further. So all of us come with nice breakfast and sit here 5 hours continuously. Continuous 5 hours, all 32 chapter revision. Is anybody thinking we should have known the details of the program during orientation only? Okay. Go. So, if you tell the story, this is how I prepared. without telling how balanced your life was. You should not tell that you play every day, you watch movie every week and all that. You should just tell how you studied and how you prepared. 11 and a half hours every day at one place, one room, no getting out of the room. Asking to go out for washroom would make us feel so awkward. The teacher will make us feel like we have asked him for his kidneys. All right. If we go 1 minute late to the study room, we would not be given breakfast the next day. All of this, when you tell, people will feel like, oh my God. That is what you should tell. You should tell like you have put in extreme hard work and you are the epitome of hard work. Then people will start relating you like, relating to you and talk about you like they talk about heart. Heart works continuously, doesn't take a break, works day and night. My daughter has worked like that. Alright, when your parents start giving that build up for you, you should just sit back and enjoy. Don't try to correct it. Let it to the imagination of people to imagine whatever they want about you when it is in the positive side. If there are things going southwards, negative side, they are making assumptions unnecessarily about you in a negative way, then you have to clarify. They are thinking that you are a super person, you are a superman, able to study 12 hours, 16 hours every day. Okay. There used to be a senior in our senior secondary school. When we were in 11th standard, there used to be one in 12th standard. He happened to be the son of our botany teacher, lady. He did well. He was preparing for medical entrance exam. Somebody started pushing rumors about him that because his scores used to be consistently high, he used to be among the toppers every time in college. So somebody started pushing the rumor that he sleeps once in 3 days. That fellow never clarified it. If anybody asks him, do you really sleep once in 3 days, he will just laugh and go. Alright? Is it practically possible? Somebody sleeps once in 3 days. I am telling you to sleep 8 hours every day. Okay, animal tissue, structural organization in animals, different types of tissues, epithelial tissue. We have simple epithelium, single layer which covers the body surfaces. Compound epithelium, multiple layers of cells, provides protection. Squamous epithelium, thin flat cells found in alveoli of lungs. Cuboidal epithelium, cube shaped cells found in kidney tubules. And again, these are examples given in NCERT only. So question will come asking you for this one. What type of epithelium is found in the tubules of kidneys? Give all the 4. If you have done it well, it's easy, you get it. Otherwise, you will start thinking and tend to do mistakes. Columnar epithelium, tall column-like cells found in the lining of intestine. Ciliated epithelium, columnar cells with cilia found in respiratory tract. Okay. Connective tissue, loose connective tissue, dense connective tissue, adipose tissue, skeletal tissue. Loose connective tissue consists of cells, fibers and ground substance. Dense connective tissue, high density of fibers, forms tendons and ligaments. Dense connective tissue. Adipose tissue stores fat found under the skin and around the organs. Skeletal tissue, bone and cartilage, provides support and structure. Muscular tissue, skeletal muscle, smooth muscle, cardiac muscle. Where is it found? What is its basic characteristic? Nervous tissue, neurons and neuroglia. Neurons are the nerve cells, conduct electrical impulses. Neuroglia are the supporting cells in the nervous system. When you go for level 1, you can write different types of neuroglia also. But are the different types of neuroglia given in NCERT? You are not given different types of neuroglia in NCERT. Don't bother to write it in these notes. Don't bother to write anything that is outside of NCERT in your linear notes. Nothing at all. In syllabus map or level 1 or perspective based notes, nothing which is not given in NCERT should be there. Alright? Because once you go down that line, you will not know where to stop. You will try to pull out from various sources and you will get stuck up with one topic and get lost. All that additional information will come in our post-test analysis notes. Only if it has come as a question in all these many years of exams that have been conducted. We are writing 2019 paper Monday. So 13, 14, 15, 16, 17, 18 over. 2019 paper we will be writing on Monday. We will be left with only 5 more papers to complete all the previous year papers of. Let us not go beyond that. I don't want to take you beyond 2013 backwards, because the format of questions in AITMT were not similar to what it is in NCRT, in NEET. So after this, I'll start giving you papers which are mock papers based on the current pattern of NEET exams. So another, this one, this Monday and 5 more Mondays. In about 6 Mondays, 1.5 months, by mid-September, not mid-September, by the end of September, you will be done with all the previous year papers. So your first target would be to complete post-test analysis of all these papers thoroughly as quickly as possible. And some of you are making mistakes in that. One is you are completing post-test analysis of maybe biology of all the paper. Physics is lagging behind, chemistry is lagging behind. Don't go subject by subject. Complete the analysis paper by paper. Alright? Paper by paper. I will give a analysis sheet for you. I have prepared it. I will get it printed and you will get it as a booklet. You will get it as booklet. Alright. The booklet will have something similar to this. Like for every paper you will have date, paper number, date, for every subject, number of questions attempted, number of questions wrong, negative marks, positive marks, total marks. And it will, and the end when you calculate, you know for the entire paper how much is what you have got. It will be printed for all the 62 tests. The booklet will have test number and the date of the test pre-entered. You have to just fill in your details, your analysis details, which you are already doing for all the other paper, which you have done for the papers, your analysis. That will be in one place for you to look back. Another one sheet will be test number, date, net score, how much is the total score you got in that. Then post-test analysis tracker. When did you start the post-test analysis of that paper? When did you complete it? How many days did you take to complete the post-test analysis for that paper? Alright? So you have to do paper by paper. Don't go by, I'll finish off biology, I'll be up to date with biology. I am lagging behind tremendously with physics. No, go paper by paper. When it comes to physics and chemistry, you can skip calculation based questions, problems. Calculation based problems which requires deeper understanding of the concepts, you can skip. Whatever is related to concepts, whatever is related to laws and rules, answer, solve all those and add it into your notes. Problem solving, we will do later stages. You should mark in every paper what are all the things that you have skipped, gap to less than 20%, excellent. By end of January to February, if you can make it up to date, very good. Alright? So don't have unrealistic expectations from yourselves. Okay? Nervous tissue. Neurons, neuroglia we discussed. Organ and organ systems. Examples of organ systems: digestive system, respiratory system, circulatory system, nervous system. Digestive system, the important organs, similarly with respiratory system, circulatory system and nervous system. Morphology and anatomy of earthworm, cockroach and frog. We don't have it in as greater detail as it used to be, but we still have it, some basic. Earthworm, segmented body. You need to understand the segments, setae, clitellum, and it's hermaphroditic. Anatomy of the digestive system only. Anatomy of the digestive system only of earthworm. Complete digestive system starts with mouth and ends with anus. Cockroach also same. Morphology is segmented body, body divided into head, thorax and abdomen. Anatomy of the nervous system. Digestive system of frog, nervous system of digestive system of earthworm, nervous system of cockroach and circulatory system of frog. That's all. Alright. Anatomy of the nervous system, brain, nerve cord and ganglia. What is special about the cockroach is this. Ganglia. Alright. Our processing happens in the center. Cockroach has a mechanism of processing throughout the body because of the ganglia. Ganglia are bundles of cell bodies. They are bundles of cell bodies in the periphery outside of the brain. In the brain also you will have bundles of cell bodies is called ganglia. In the brain the same bundles you call it as nucleus. In the brain you call it as nucleus, outside of the brain you call it as ganglia. We also have ganglia, we have sympathetic ganglia. Alright, in the sympathetic nervous system, sympathetic chain we call it. Multiple levels of sympathetic ganglia we also have. So frog morphology is amphibious, moist skin. Body divided into head, trunk and there is no tail. No tail throughout its life cycle. Frog? No tail throughout its life cycle? Tail used to be present during the tadpole stage. 3-chambered heart, closed circulation. Then cell theory, historical background, contributions by Schneiden, Schwann and Virchow. Schneiden gave details about plant cells, Schwann gave details about animal cell, Virchow gave details about cell division. Why important? These fellows have been referred to repeatedly. Questions have come about their contribution. Like you have to remember Mendel is referred to as the father of genetics. If a question comes on that, we should be able to answer that. So these some names associated with some discoveries we have to remember, and we will also limit that as well to NCERT. Modern understanding, all living organisms are composed of cells. All cells arise from pre-existing cells. This is the key principles of cell theory. These are the key principles of cell theory. Definition, cell is the fundamental structural and functional unit of life. Cells are the basic units of life in all living organisms. Types of cells, prokaryotic and eukaryotic. Prokaryotic, no nucleus, eukaryotic, nucleus and organelles. Structure, basic components of a cell. Plasma membrane, cytoplasm and nucleus. Those are the basic compartments, if you can say. Boundary is the plasma membrane, everything is nucleus, everything is cytoplasm. In the center, specialized is nucleus. Prokaryotic cell characteristics, small, lack membrane bound organelles, DNA is not enclosed within the nucleus. Cell envelope, complex structure with glycocalyx, cell wall and plasma membrane. This is important. Glycocalyx is what will form the slime layer or capsule around bacteria. Bacteria with capsule are gram-positive bacteria or gram-negative bacteria. Bacteria with capsule are gram-positive or gram-negative. Gram-positive. Sure? Gram stain is what? What is gram stain? What color? Color, what color do you choose for gram stain? Huh? Blue color. Blue. My daughter is 2 years old. Alright. She likes colors. She identifies colors. She can recognize more colors than I can. I have problem with recognition of colors. I can see the colors, but I cannot tell what color it is. Okay. Blue, she says, for blue. Orange, for orange. So in gram staining, you use blue and orange. Okay. Blue is the primary stain. Orange is the secondary stain. And primary stain is for cell wall. Primary stain is for cell wall. Cyanin. Alright. If the bacteria which take up the primary stain are gram positive. Those which do not take up the primary stain are stained with secondary stain, which is safranin, which will stain them orange. They are gram negative. Now tell me, if a bacteria has capsule around it, will it be gram positive or gram negative? Gram negative, because the capsule will cover off the cell wall. It is outside of the cell wall. If there is a capsule, the capsule of glycocalyx. Glycocalyx is made up of two chemicals, N-acetyl glucosamine and N-acetyl muramic acid. N-acetyl muramic acid and N-acetyl glucosamine. So they will not allow the pigment to cross the capsule and stain the cell wall. So cell wall doesn't take up the stain. So they are gram negative. Gram negative bacteria are more dangerous infection wise. So everything correlating one to each other. See now. He's looking around to see. All right. So all that about the bacteria, the cell structure correlates. Correlation will happen when we have the references, no? So this is important. Ribosomes, 70S ribosomes. Prokaryotic cells have 70S ribosomes. They are the site of protein synthesis found in the cytoplasm. 70S ribosome components are 30S and 50S. But 30 plus 50 is 80, no? Why 70? You have a 20 rupee note and a 50 rupee note. Totally you have 60 rupees. You have a 30 rupees, you have 30 rupees and 50 rupees. What is the total you have? 70 rupees. Is that how it is? Then why 70S? Sedimentation coefficient, but how? Huh? It settles up to the level of? At 70S, correct. Our business tycoon is looking at me like, what nonsense are you asking? Isn't it so obvious? It settles at 70S. There is overlap of the 30S and 50S ribosomes. 30S and 50S ribosomes overlaps. So when you take the component and allow it to sediment in a tube, sedimentation tube, the smaller unit of the ribosome will sediment up to 30 units. So you call it 30S ribosome, 30 units on the sedimentation coefficient. The bigger unit will sediment up to 50 units. But when you put the complex ribosome, both the smaller unit and the bigger unit together, because of the overlap, the space each unit occupies is not the same as the total of both the units separately. Alright, if you take separately 30S and 50S separately and allow it to sediment in the same tube, it might sediment to the 80S only or even more, because there is more space created between the sedimenting ribosomes. The unit completely sediments up to 70 units. The complete ribosome sediments up to 70 units, that is 70S. The ribosome in eukaryotes is 80S. It is 40 plus 60. 40, 60. Inclusions are there. Reserve materials like phosphate granules, glycogen granules, they are all stored in the cytoplasm in the prokaryotic cell. Eukaryotic cell, membrane bound organelles, well defined nucleus, found in protista, plants, animals, fungi, everybody else except bacteria. Cell membrane, phospholipid bilayer, the fluid mosaic model. Fluid mosaic model is important. The questions will come on the fluid mosaic model. Semi-permeable controlled substance movement. Cell wall found in plants, algae and fungi. Provide shape, protection and support. Endomembrane system. Endomembrane system. Question. Which of this is not a part of the endomembrane system is a question. Have you seen that question in any of the papers? Endoplasmic reticulum, Golgi complex, lysosomes, vacuoles. Mitochondria is also a double membraned organelle. But why is it not a part of the endomembrane system?
Because its mitochondria's function is not coordinated with this.
All of these are in continuity. There is nuclear membrane, continuing with that is endoplasmic reticulum, then Golgi complex coordinates with that, lysosomes and vacuoles come out of Golgi complex. So they are all functioning as a unit in decoding the code, final processing and packaging, and the packets. Decoding the code is in the endoplasmic reticulum. Final processing and packaging happens in the Golgi complex. Packets are lysosomes and vacuoles. Lysosomes and vacuoles or vesicles are all packets. Continuity, it's a factory. Endomembrane system is a metabolic factory within the cell. Mitochondria is independent of this. Correct. So that is why it is not considered a part of the endomembrane system. Question has come. All these four: endoplasmic reticulum, Golgi complex, lysosome, mitochondria. Which of these four is not a part of the endomembrane system? Plastids, chloroplasts, chromoplasts, leucoplasts. Again here, what pigments are commonly present? Here no pigment. Chromoplasts will have phycoerythrin, phycocyanin, different colored pigments. Chloroplasts will predominantly have chlorophyll. Ribosomes, ATS ribosomes found in the cytoplasm and on rough endoplasmic reticulum. Cytoskeleton, network of protein fibers including microtubules, microfilaments, intermediate filaments. These three are important. Microtubules, microfilament and intermediate filaments. What is their structure and where are they useful? Microtubules and microfilaments are useful in cell division, aster formation, spindle fiber formation. They are all the cytoskeletal elements only, and intracellular transport is happening because of these fellows. They are contributing to the movement of material from one part of the cell to another part of the cell. It is not like the cell will turn and what is here comes here, and if it has to transport it backwards, it will turn again. No, it is not because of gravity. It is because of controlled movement. It is delivery. There is a entire logistic system running inside the cell. Logistics is movement of material. Alright. Cilia and flagella, hair-like structures involved in cell movement. Nucleus contains genetic material, controls cell activities, site of RNA synthesis. Chromosomes, structures within the nucleus containing DNA, involved in heredity and cell division. Microbodies are vesicles containing enzymes, involved in various metabolic activities. Are microbodies permanent organelles inside the cell? Are microbodies permanent organelles within the cell? Microbodies are temporary structures. They are produced as and when required. You can produce any number of microbodies based on the requirement of the cell. So therefore microbodies should not be considered as organelle ecosystem of the cell. Done. Next is biomolecules. We did 8 chapters. 8 chapter revision in 3 hours. Okay. So I'll give you this. Before I come next time, that is 7th of September, we will have this. I have changed the schedule a little bit so that you will not have absence of senior faculties for more than 2 days for the rest of the duration. Alright. So you will have continuously some presence of some senior faculty. The cycle continues. Alright. I am very conscious of what is going on and what needs to be changed, what needs to be modified. All of that will be happening from time to time. If there is anything that you feel is hindering your progress, you should let me know. You can tell your resident faculties. They will communicate it and we can bring about whatever necessary changes that need to happen from time to time. Alright. Otherwise just keep going. Alright. Okay. Any questions? Anything that anybody wants to ask? Alright. Are you all playing well? If there is no rain or anything, if you are playing well, physical activity is happening properly? increase during the morning session, like 5:15 to 6 o'clock. I'm sure all of you have become comfortable with 12 Surya Namaskaras. Increase it. Increase it for the next month, make it 15. Add 3 more. Your body needs to be challenged. Surya Namaskaras, your body needs to be challenged. I don't want to add more and more asanas to it. Surya Namaskara itself is a very balanced cycle of yogic postures which will do great benefit to your body and the mind also. So gradually bring it up to 25. Joint mobilization, 25 Surya Namaskaras and 10 minutes of breath control, breathing exercises, 5 minutes of meditation should settle it down. You have to make it faster also. Don't run away from that, okay? You will thank me later. You become models and actors and superstars in your colleges. Even if you don't become professionally models and actors, you will be appreciated for your physical appearance if you do Surya Namaskara in your college. Alright? All of that will add to the quality of life you will have in medical college. Alright? So, but again, remember, whatever of these small tidbits that I am giving you will translate into big money for you. Don't forget me. Alright? Okay. Make big money. All the best. See you all shortly in my next visit. Keep working. I'll be leaving today. Post lunch by around 2 o'clock, 2:30 I'll leave. Alright?
On Sundays, can you spend more time with our parents?
Parents. Good. You raised it. Alright. I am getting reports that your parents are allowing you to connect your tabs to their Wi-Fi hotspot in their phones and you are downloading things. You know who leaked this information? Parents. See a few songs that you might like to listen to while you sleep and rest. All that is fine. But don't download movies to watch. Don't download any visual content on your tabs. Alright? It is possible to check each one of your tabs. It is possible because only 48 of you are there. Randomly your tabs can be picked up by resident faculty and the faculties and the warden, and all of your tabs can be checked for the content that is present on the tabs. That is possible. It is not a difficult thing to do. But I don't want to do that unless it becomes necessary. So exercise that restraint. Only those, maybe if you are a music person who loves to listen to music while you rest, it will let you rest well. If music can help you improve the quality of your rest, take only that kind of music from your parents and use it only during rest without disturbing others. No other medium. Alright, no other series, movies, no. Have this, enjoy this phase. Sundays, all of you have given your recommendation of movies, what movie you want to watch. And randomly some movie gets selected by Chit. It is fun. Be in the present, go with the flow, have fun with this process. Alright? Don't break this isolation. Don't break the isolation. Alright? Isolation will lead to specialization. Don't break this isolation because you are comfortable in whatever you are comfortable with. You have to break that. No, not during the study hours. During those 11.5 hours, baseline for that is you can do all that which you are allowed to do in an examination hall. While writing the exam, what all is allowed in this room. during the 5 scheduled sessions. Outside of the 5 scheduled sessions, you are not supposed to be in this room. Go play. 4 o'clock to 5 o'clock, you cannot say, I don't want to play, I will sit and study. No, go play. Do what you are supposed to do at the respective times. Alright? No music during studies. No. It might be a habit. You might have felt good while studying, listening to music, but are you allowed to do that in the exams? No. So don't do it. Then your mind will want music to perform better during exams also. Okay? Anything else? The question she asked led me to redefining certain ground rules. The question is, can we spend more time with parents on the days when they visit us? You can. 2 to 4 is your free time. You can spend time with them. 5:30 is the movie. So basically you have 2 to 5:30. 2 to 5:30 is the time you have that you can spend with parents. But you cannot go out of the campus. They cannot take you outside of the campus. Probably the scope of coming in your room and staying with you also is limited. Correct, no? Can they come and stay? You barely have space for sleeping. Correct, no? It is just like our rooms are just like train compartments. You can only sleep in your respective berths. Where will you accommodate your parents also? Campus is big, you can sit around, you can talk, all that is fine, no problem, you can spend time. 5:30, come back for the movie. So 2 to 5:30. Or even it is like 12:30 the session is over. 12:30 to 5:30 is the time, technically speaking. Isn't it? You can tell your parents to come around 12:30 also. They can join you for lunch. Is it possible, madam? They can join for lunch? Parents? We don't know that. Alright. Let's see. Because that will require some logistic arrangements. That will require a lot of other changes to be made. So around that time they can come. Around 12:30, 1 o'clock if they come, you can spend time with them till 5:30. Don't skip the movie. Alright? Don't say I will not watch the movie, I will spend time with parents. Watch the movie also. And don't select all motivational movies. Select out and out entertainment movies. Complete entertainment. All nonsensical entertainment, masala movies. Alright? Okay. Alright, you can leave. All the best. Stay on track. Stay happy.